Right, so like pop quiz, everyone. Just kidding. But get ready, because today we're diving deep into chromosomes and heredity.
Heredity.
Yeah, it's like biology class, but way more fun, hopefully.
Yeah.
Um, so have you ever like wondered how you got your mom's eyes or your dad's height or even like
Yeah, like why some people are colorblind.
why some people are colorblind?
It's
Exactly.
all about those genes.
It is all about the genes, like those
Wow.
tiny little blueprints that make you.
Yeah, and those genes, they live on chromosomes, right?
They do. They live on
So
chromosomes.
like instruction manuals passed down from your parents.
Exactly, like little instruction manuals.
Okay, so let's start with the OG of genetics.
Mendel.
Mendel, the peat plant guy, right?
Yes.
What did he do that was like so groundbreaking?
Well, Mendel, he was really a meticulous observer.
Yeah, back in the 1800s.
Way back in the 1800s, yeah.
Like no fancy equipment.
No fancy equipment.
Yeah.
He just noticed these patterns in how traits like flower color or seed shape were passed down through generations of pea plants.
And he called these units of inheritance factors.
Factors? Okay.
And even though he didn't know about DNA or chromosomes, he figured out the basic rules of inheritance.
So he was basically doing genetics before genetics was even a thing.
Exactly. He was ahead of his time.
Wow. So how did scientists later connect like Mendel's factors to chromosomes?
Well, it was around the turn of the century, scientists like Sutton and Boveri,
Okay.
they were studying how cells divide.
That's that process called meiosis.
That's meiosis, exactly. And they noticed that the way chromosomes behave during meiosis really mirrored Mendel's factors.
Oh, so like chromosomes, they pair up.
They pair up,
They swap bits.
swap bits.
And then they separate into new cells, just like those traits that Mendel was tracking.
Exactly like those traits Mendel was tracking.
It's like those aha moments in a detective movie.
It is.
Like when all the clues finally click.
It all comes together.
So that's what led to the chromosome theory of inheritance.
That's right. The chromosome theory of inheritance.
So the idea that genes are actually located on chromosomes.
That genes are on chromosomes, yes.
Imagine being a scientist back then, like making that connection.
It must have been mind-blowing.
Right. So let's talk about something that I think is really fascinating.
Okay.
Sex-linked inheritance.
Sex-linked inheritance.
What is that all about?
Well, it just means that certain traits are passed down differently depending on whether you're male or female.
Ah. Now,
Yeah. And this is because the genes for these traits are located on the sex chromosomes X and Y.
okay, so those chromosomes that determine if you're biologically male or female.
Exactly.
But why are most sex-linked traits on the X chromosome?
That's a great question. Well, think about it. The Y chromosome.
Yeah,
It's much smaller than the X.
it goes smaller, right?
Way smaller, like a tiny instruction manual.
Compared to a big textbook.
Compared to a big textbook,
Okay, so there's just less room for genes.
less room for genes on the Y chromosome.
So what's a good example of this sex-linked inheritance?
Well, colorblindness.
Oh yeah, color blindness.
That's a classic example. It's a recessive trait, and it's on the X chromosome.
Okay.
So this means that females need to inherit 2 copies of the colorblindness gene, one from each parent to be colorblind.
Okay.
But males, they only need 1 copy because they only have 1 X chromosome.
That's why it's more common in males.
Exactly.
Like they just need one hit from the gene to have it.
1 hit and they've got it.
Wow. Okay, so let's fast forward a bit to the early 1900s.
Okay, early 1900s.
And meet Thomas Hunt Morgan,
Thomas Hunt Morgan.
the fly guy of genetics.
The fly guy.
Didn't he find something that like challenged Mendel's ideas?
He did. He was breeding fruit flies and studying their eye color, expecting to see the classic Mendelian ratios.
Okay.
But then, in a sea of red-eyed flies, a white-eyed male popped up.
A white-eyed male.
This little fly threw a wrench into things.
So like a white-eyed fly.
White-eyed fly.
Why was that such a big deal?
Well, remember, Mendel saw traits being passed down in these specific ratios.
Yeah.
But when Morgan crossed these 2 red-eyed flies, the white-eyed male offspring didn't fit the expected pattern.
Oh.
And then when he crossed that white-eyed male with a red-eyed female,
Okay.
he couldn't get a female with white eyes no matter what he tried.
So it's like Mendel's laws were working, but only sometimes.
It was really puzzling.
So what did he do?
He dug deeper. He did tons of experiments and careful observation.
Okay.
And then he proposed this radical idea that the gene for eye color in fruit flies must be located on the X chromosome.
On the X chromosome.
On the X chromosome.
Wow.
So he proved that genes have an address on chromosomes.
An address of physical
A
address.
full address.
But how did he know It was on the X and not just like floating around.
Oh, it all came down to the inheritance patterns. Those white eyes showed up almost exclusively in males.
Yeah.
And remember, males only have one X chromosome.
Right.
So they inherit that gene for white eyes, bam, they've got white eyes.
Bam, white eyes.
But females, they need 2 copies to express that trait.
So that's why he couldn't get a white-eyed female from that cross. The females, they always got at least one red-eye gene from their moms,
At least one red eye gene.
masking that white-eye gene.
Masking the white eye gene.
Uh-huh. So how do scientists actually track and predict these sex-linked inheritance patterns?
Well, that's where Punnett squares come in.
Punnett squares?
Those handy little glids.
Like from high school biology?
Exactly. From high school biology, we use those to predict the outcome of these genetic crosses.
Right, right.
And we can adapt them to handle sex-linked traits, too.
Okay, so we're still using those squares.
Still using the squares.
But now we're factoring in the X and Y chromosomes.
Factoring in X and Y.
Got it.
It helps us predict the offspring's genotype, their phenotype, and even their sex.
So it's like adding another layer of information?
Another layer of information.
Okay, but here's something I've always wondered.
Okay.
Females have 2 X chromosomes,
2 X chromosomes.
while males have just 1.
Just 1.
Don't both sexes need the same amount of proteins produced from those genes on the X chromosome?
That is a fantastic question.
Like, how does that work if females have double the genes?
Well, you've stumbled upon one of the coolest tricks in genetics. X chromosome inactivation.
X chromosome inactivation sounds intense.
It is pretty cool.
Okay.
So think of it like this: early in development, one of the 2 X chromosomes in each female cell randomly gets switched off.
Switched off, like turned off.
Turned off. Basically, most of the genes on that chromosome go silent.
So even though females have 2 X chromosomes, only 1 is fully active in each cell.
It's like having a backup generator.
Okay.
You only need one running at a time.
Okay.
And this process makes sure that females don't produce double the amount of protein from X-linked genes.
Wow, genetics is way more dynamic than I thought.
It really is.
So is there a way to actually see this X chromosome inactivation happening?
Have you ever seen a tortoiseshell cat?
Tortoiseshell cat.
Those beautiful felines with patches of black and orange fur?
Oh yeah, they're gorgeous.
They are.
What do cats have to do with X chromosomes?
Well, the gene for fur color in cats is on the X chromosome.
Okay.
And tortoiseshell cats are heterozygous, meaning they have one allele for orange fur and one for black fur.
Okay.
And remember, X inactivation,
It's random.
it's random.
Yeah.
So in some cells, the X with the orange allele is active,
Okay.
resulting in orange fur.
Orange fur.
In other cells, the X with the black allele is active, giving black fur.
So their fur is like a map
It's like a map
of which X chromosome is active
of which X chromosome is active
in each cell.
in each cell.
That's amazing. I never knew cat fur was so genetically complex.
It's a visual representation of genetics in action.
So we've been talking about traits that are controlled by one gene, but what about traits like height or skin color?
Right. Those are great examples.
They come in so many variations, not just 2 or 3 options.
You're right. Those traits are controlled by multiple genes working together.
Okay.
We call this polygenic inheritance.
Polygenic inheritance. So it's not just one gene calling the shot. It's like a whole team effort.
Exactly. A whole team effort.
Okay.
Each gene contributes a little bit to the final trait.
Yeah.
And because there are so many genes involved, the possibilities for variation, they skyrocket, creating that spectrum of phenotypes we see.
Okay, so the more genes involved,
More genes involved.
the more subtle the variation.
Exactly. It's not just tall or short.
Right.
It's a whole range in between.
Is there an example that really shows this continuous variation?
Well, think about ear length in corn.
Ear length in corn.
You can have ears that are long or short,
Okay.
or somewhere in between.
Like a gradient.
It's like a gradient, not just 2 distinct categories.
Okay.
And it all boils down to those different combinations of genes and their alleles.
So are all genes created equal in terms of how much they influence a trait?
That is another great question, and the answer is no.
Okay.
Not all genes have the same power.
Okay.
Some act as modifiers, tweaking the expression of other genes.
Modifiers, so like they're fine-tuning the orchestra.
Exactly, like fine-tuning the orchestra.
They don't play the main melody,
Right.
but they add depth and nuance.
They add all the richness.
Okay, so give me an example.
We'll take eye color, for example.
Eye color.
It's not just brown or blue. There's green hazel,
Yeah.
even shades of gray.
Right, there's a whole rainbow of eye colors.
All rainbow of eye colors.
How does that work?
Well, the main gene for eye color determines whether you have pigment or not.
Okay.
Modifier genes can influence how much pigment you have
Okay.
and how it's distributed.
Okay.
And that creates all those variations we see.
So the main gene sets the stage.
Sets the stage.
And then the modifier genes add those like special effects.
The special effects.
Cool.
Exactly.
Wow, this is remarkable.
It is.
It seems like every time we peel back one layer of genetic complexity,
Every time we peel back a layer,
there's another one waiting to be discovered.
there's always more to explore.
So what's another layer?
Well, one of those fascinating layers is gene imprinting.
Gene imprinting.
Gene imprinting.
That sounds like something straight out of a sci-fi movie.
It does sound a little sci-fi.
What's that all about?
It's a process where certain genes are tagged with these chemical markers
Okay.
that can activate or silence them. So it adds another layer of control over gene expression.
So it's like some genes are pre-programmed
Pre-programmed,
to be on or off,
on or off,
depending on which parent they came from.
depending on which parent they came from.
Wow.
It's another complexity.
That's so cool. My brain is officially full.
Full of genetics.
This deep dive into chromosomes and heredity has been amazing.
It has been fun.
It's incredible how much we've learned about genetics, and it's exciting to think about all the discoveries yet to come.
Oh, absolutely. There's so much more to learn.
So what stands out to you as the most surprising or interesting part of what we talked about today?
I think for me, it's the sheer complexity of how genes are regulated, like X chromosome inactivation.
Yep.
It's this mind-blowing mechanism that ensures that both males and females have the right amount of X-linked gene products, even though they have different numbers of X chromosomes.
Right.
It's a testament to how intricate and balanced our biology is.
I agree. It's a remarkable example of how finely tuned those life processes are.
Exactly.
And seeing that process like reflected in the fur of tortoiseshell cats.
Oh, yeah.
It's just incredible.
It's amazing.
It's a reminder that genetics isn't just abstract theory.
It's not just theory.
It's literally shaping the world around us.
It is shaping the world around us. That's right. We've got a lot more to uncover in the world of genetics.
Okay, all right.
So let's dive into this world of personalized medicine and gene editing.
Personalized medicine and gene editing.
These fields are revolutionizing healthcare.
Yeah, and it all stems from the amazing discoveries we've been talking about.
Exactly.
Okay, so first up, personalized medicine. What's the big idea behind that?
Well, it's all about using your unique genetic information
Okay.
to make healthcare decisions tailored just for you.
So it's like moving away from that one-size-fits-all approach
Exactly.
and recognizing that everybody's body responds differently to treatments based on their genes.
Precisely.
So instead of everyone getting the same medication for a condition, doctors could use genetic tests to figure out which drug would work best for each patient
Exactly.
and which ones might cause side effects.
Right. It's all about optimizing that treatment by really understanding your genetic predisposition.
So a drug that's a miracle for one person could be a dud or even dangerous for someone else, depending on their genes.
Exactly. And that's where personalized medicine steps in.
Wow.
By knowing your genetic profile, doctors can choose the right treatment for you, maximizing those benefits and minimizing the risks.
That's a game changer.
It really is.
Okay, so let's talk about gene editing.
Gene editing.
And that sounds straight out of a sci-fi movie.
It might sound futuristic,
Yeah?
but it's becoming more and more real every day.
Wow.
Gene editing is a powerful tool that lets scientists actually change the DNA sequence of genes.
So we could actually correct genetic defects that cause diseases.
That's the goal. Imagine editing out the mutated gene that causes cystic fibrosis or sickle cell anemia.
Wow.
It could revolutionize how we treat genetic diseases and maybe even cure them.
It's mind-blowing to think that we might be able to go right to the source of the problem and fix it at the genetic level.
I know, it's really exciting.
But I imagine there are some ethical considerations that come with such a powerful technology.
Oh, you're absolutely right.
Yeah.
Gene editing raises some serious ethical questions about responsible use.
Right.
Should we only use it for serious diseases, or is it okay to enhance traits like intelligence or athletic ability?
Yeah, like where do we draw the line?
Exactly, where do we draw the line? It's a big question.
So how do personalized medicine and gene editing connect back to our understanding of chromosomes and heredity?
Well, personalized medicine and gene editing wouldn't even be possible without that deep understanding of how genes work, how they're passed down, and how they affect our health and traits. Without that knowledge, we wouldn't be able to interpret genetic tests, identify those disease-causing mutations, or even dream about editing genes.
So all those discoveries we talked about in part one, from Mendel's pea plants to Morgan's fruit flies, they laid the groundwork for these cutting-edge advances in healthcare.
Absolutely. Mendel's laws of inheritance gave us the basic rules of the game, showing us how traits are passed down from generation to generation.
Yeah.
Morgan's work on sex-linked inheritance revealed how genes are arranged on chromosomes and how they separate during meiosis.
Right, right.
And then the discovery of DNA is the blueprint of life that really unlocked the secrets of the genetic code itself.
It's amazing to see how scientific progress builds on itself. Like, each discovery leads to even more incredible breakthroughs.
It really is like a giant puzzle
Yeah.
where each piece we find helps us see the bigger picture.
And it's a testament to human curiosity
It is.
and our drive to understand the world around us.
I agree. It's what makes science so exciting.
That there's always something new to discover.
Always something new to discover.
More mysteries to unravel.
More mysteries to unravel.
More ways to use our knowledge to make a difference.
Exactly. And it's a reminder that even seemingly small discoveries like those white-eyed fruit flies
Right.
can have a huge impact on our understanding of the world and lead to innovations that benefit everyone.
So speaking of innovations, what are some of the amazing tools and technologies that scientists are using to study genetics today?
Oh, we've come a long way from peering through microscopes.
I bet.
We've got high-powered microscopes that let us see individual chromosomes,
Wow.
DNA sequencing machines that can read the entire genetic blueprint of an organism.
That's incredible.
Technology has revolutionized the way we study genetics.
I remember reading about something called PCR,
PCR,
polymerase chain reaction.
polymerase chain reaction.
That really changed the game.
It really did. PCR is a workhorse in the lab.
Okay.
It acts like a molecular copy machine, allowing scientists to amplify tiny bits of DNA into millions of copies.
So with PCR, scientists can work with even the tiniest samples of DNA.
Exactly like from a crime scene or a fossil or even a single cell.
Wow.
It's got applications in so many fields: research, medicine, forensics, you name it.
Wow.
It's a perfect example of how technological advances have really propelled our understanding of genetics forward.
What about DNA sequencing?
DNA sequencing?
It seems like there are always news stories about breakthroughs in sequencing technology.
Yeah, DNA sequencing is another groundbreaking tool that's transformed genetics.
Mhm.
It allows us to determine the exact order of nucleotides in a DNA molecule.
So with DNA sequencing, we can read the instructions that make up an organism's entire genetic blueprint.
Precisely. And the speed and affordability of DNA sequencing have improved dramatically in recent years.
Really?
We've gone from sequencing the first human genome, which took over a decade and cost billions of dollars,
Yeah.
to sequencing an individual's genome in a matter of days for just a few $1,000s.
That's incredible progress.
It is incredible progress.
What does that rapid advancement mean for our understanding of genetics and its applications?
Well, it's opening up a whole new world of possibilities.
Yeah.
With DNA sequencing becoming more affordable and accessible, we can gain deeper insights into the genetic basis of diseases, develop personalized treatments, and even trace our ancestry with greater accuracy.
So it's not just about understanding ourselves better.
It's not just about us.
It's about unlocking the secrets of our past
Unlocking the secrets of our past
and shaping the future of healthcare.
and shaping the future.
It seems like the future of genetics is incredibly bright.
It is incredibly bright. DNA sequencing is a powerful tool that's changing how we understand life itself.
But even with all these incredible advances,
Yes.
it's important to remember that we've only just begun to explore the vast complexities of the genome.
We're just scratching the surface.
So true. There's still so much more to discover and understand about our genes and how they shape who we are.
Yeah, it's amazing to think that each of us carries this incredible genetic blueprint inside every cell of our bodies.
I know, it's like a story.
That's been passed down through generations, shaping who we are today.
Right, and it's not just our own individual story. It's the story of life itself.
That is the story of life itself.
From the simplest single-celled organism
Yeah.
to complex creatures like us, we all share those same basic building blocks of DNA and chromosomes.
Ew, it's pretty amazing.
It's mind-blowing to think about the incredible diversity of life on Earth.
I know.
From tiny bacteria to giant sequoia trees
Yeah.
and everything in between, all arising from those same fundamental genetic components.
It's really awe-inspiring, that diversity.
It is.
And it all comes down to the way those genetic building blocks are arranged and expressed.
Right.
Each species has its own unique set of genes and variations within those genes that give rise to its distinctive traits.
It's like nature's a master artist,
Yeah.
constantly experimenting with different combinations of colors and brushstrokes to create this masterpiece of biodiversity.
I like that analogy.
And that process of experimentation driven by mutation and natural selection.
Yes.
That's what we call evolution.
That's evolution.
It's the force that has shaped the incredible tapestry of life we see today.
It's incredible to think about.
Thinking about the vast stretches of time over which these evolutionary processes have been operating, shaping and reshaping life on Earth.
Yeah.
It's truly humbling.
It puts our own existence into perspective, doesn't it?
Yeah, it does.
We're part of something much larger than ourselves, connected to all living things through this shared genetic heritage.
This has been an incredible journey through the world of chromosomes and heredity.
It has.
We started with Mendel's pea plants
Yeah.
and ended up exploring cutting-edge technologies like gene editing. It's amazing to see how far we've come in our understanding of genetics.
We've covered a lot of ground,
We have.
from those basic principles of inheritance
Right.
to the complexities of gene expression,
Yeah.
and even those ethical considerations that come
Yes.
along with our growing knowledge.
It's clear that genetics is a rapidly evolving field with new discoveries happening all the time.
All the time, yeah.
So what do you think is the most important takeaway from our exploration?
Well, I think for me, it's the interconnectedness of it all.
Okay.
From Mendel's simple experiments with pea plants to the development of personalized medicine,
Right.
we've seen how each discovery builds upon the last, leading to a deeper understanding of life itself.
And that understanding has the power to transform our approach to healthcare agriculture
Yeah.
and even our relationship with the natural world.
Absolutely.
It's a beautiful reminder that science is not just a collection of facts,
It's not.
but a dynamic and ever-evolving process of discovery.
It's a journey that's constantly unfolding, leading us to new insights and possibilities.
I couldn't agree more so as we wrap up this deep dive.
Okay.
What final thought would you like to leave our listeners with?
Well, our exploration of chromosomes and heredity has shown us those intricate mechanisms that make life possible, but it's also revealed those profound connections we share with all living things. So let that knowledge inspire you to keep learning, keep questioning, and keep marveling at the wonders of the natural world.
That's a perfect note to end on, listeners. We hope you've enjoyed this deep dive into the fascinating world of chromosomes and heredity.
It's been fun.
Remember, this is just the beginning of your genetic journey.
Yeah.
Keep exploring, keep asking questions, and who knows, maybe one of you will make the next groundbreaking discovery in genetics.
Maybe so. Until next time, keep on learning.